Hello, my name is Dan Brimberry, and I'm a clinical researcher at the Cedars-Sinai MAST Program, and I'm going to present my poster. The bacterial composition of a healthy gut microbiome remains elusive, but multiple factors contribute. Currently, what little is known about the human gut microbiome close to death is based upon stool analysis, which is unrepresentative of the small bowel. The goal of this study is to assess the small bowel microbiome of patients who are living at the time of study participation and later died from any cause, compared to currently living subjects in the reimagined study. Antemortem subjects were identified in the reimagined cohort and compared to currently living controls. Antemortem subjects were then subsequently matched one-to-one to an individual currently living subject by age, body mass index, and sex in a confirmatory subanalysis. There were no significant differences in alpha diversity for either comparison. Beta diversity was only significantly different comparing the Antemortem cohort to the entire cohort of currently living controls. At the genus level, 129 genera were different for the Antemortem cohort compared to the entire cohort. 71 genera were different for the Antemortem cohort compared to the matched cohort. When comparing the Antemortem cohort to the entire cohort of currently living controls, the relative abundances of hydrogen sulfide-producing genera Disulfovibrio and Fusobacterium, as well as the histamine-producing genus Rautella, increased. When comparing the Antemortem cohort to matched controls, the relative abundances of gram-negatives Aeromonas and Citrobacter, the latter a hydrogen sulfide producer, increased. Similarly, both Lactobacillus, a small bowel disruptor, and Bacteroides, a genus commonly found in the stool microbiome, were both increased. Lastly, SIBO was more common in the Antemortem group compared to the entire cohort of currently living controls, and when there was growth on either McConkey or blood agar, there appears to be a relationship between colony count when upper endoscopy is performed closer to death. This is the first study to examine the microbial changes in the small intestine in pre-deceased subjects. There is an increase in microbial loads as upper endoscopy is performed closer to death, as well as an increased relative abundance of organisms that produce hydrogen sulfide and are fecal in nature. Thank you for visiting our poster at DDW 2025.